A transglutaminase is an enzyme that catalyzes the formation of an isopeptide bond between a free amine group and the acyl group at the end of the side chain of protein or peptide-bound glutamine. The reaction also produces a molecule of ammonia. Such an enzyme is classified as EC2.3.2.13. Bonds formed by transglutaminase exhibit high resistance to proteolytic degradation. Transglutaminases were first described in 1959. The exact biochemical activity of transglutaminases was discovered in blood coagulation protein factor XIII in 1968. Physiological transglutaminases, 8 transglutaminases have been characterized. Mechanism of action: transglutaminases form extensively cross-linked, generally insoluble protein polymers. These biological polymers are indispensable for the organism to create barriers and stable structures. Examples are blood clots, as well as skin and hair. The catalytic reaction is generally viewed as being irreversible, and must be closely monitored through extensive control mechanisms. A collection of the transglutaminase substrate proteins and interaction partners is accessible in the Transtab database. Role in disease: deficiency of factor XIII predisposes to hemorrhage. Concentrated enzyme can be used to correct the abnormality and reduce bleeding risk. Anti-transglutaminase antibodies are found in celiac disease and may play a role in the small bowel damage in response to dietary gliadin that characterizes this condition. In the related condition dermatitis herpetiformis, in which small bowel changes are often found and which responds to dietary exclusion of gliadin-containing wheat products, epidermal transglutaminase is the predominant autoantigen. Recent research indicates that sufferers from neurological diseases like Huntington's and Parkinson's may have unusually high levels of one type of transglutaminase, tissue transglutaminase. It is hypothesized that tissue transglutaminase may be involved in the formation of the protein aggregates that causes Huntington's disease, although it is most likely not required. Mutations in keratinocyte transglutaminase are implicated in lamellar ichthyosis. Industrial and culinary applications. In commercial food processing, transglutaminase is used to bond proteins together. Examples of foods made using transglutaminase include imitation crab meat, and fish balls. It is produced by Streptobacillium obereans fermentation in commercial quantities or extracted from animal blood, and is used in a variety of processes, including the production of processed meat and fish products. Transglutaminase can be used as a binding agent to improve the texture of protein-rich foods such as sumi or ham. Molecular gastronomy. Transglutaminase is also used in molecular gastronomy to meld new textures with existing tastes. Besides these mainstream uses, transglutaminase has been used to create some unusual foods. British chef Heston Blumenthal is credited with the introduction of transglutaminase into modern cooking. Wiley Dufresne, chef of New York's avant-garde restaurant WD-50, was introduced to transglutaminase by Blumenthal, and invented a pasta made from over 95% shrimp thanks to transglutaminase. See also, sumi, boneless fish. References. Additional sources Kellier, James B. Industry defends ingredient critics deride as meat glue. Chicago Tribune. Retrieved July 20, 2012. U.S. Patent 5,156,956 A transglutaminase catalyzing an acyl transfer reaction of AI carboxyamide group of a glutamine residue in a peptide or protein chain in the absence of CZ2+. External links: Transglutaminase,